Hi guys. So this is the last of the 3 subtopics that relate to phylogenetic disagreement. And so what we're going to talk about is a couple of different tests that you can use to figure out whether one data set perhaps is better than another data set, or you can use another test to figure out if you should maybe combine your data sets. And so what do systematists do when different data sets yield different phylogenies? And that's pretty common. And so we're going to talk about 2 different tests. But before we get to that, I want you to review a couple of different concepts. Also, this slide has a picture of a sea lion or a seal on it. I want you all to put on your thinking caps or just guess. And when we meet in class on Wednesday during our group chat, tell me whether this is a sea lion or a seal and why. And the only reason I have this on here is because we're going to talk about carnivores, the carnivore data set. So let's get started. The first thing that I want you all to remember is that, and there's a list of 4 things that I put on here, is that systematists can look at different data sets, right? Different types of characters. And so we're going to look today at some examples where you're maybe looking at morphology and molecules of the same group of taxa. So you can use morphology, molecules, which includes DNA, RNA, protein, and you can also look at things like behavior or perhaps larval characters as opposed to adult characters of a certain taxon set. Also, think about how scientists coin or think about null versus alternate hypotheses. What is a null hypothesis? What is an alternate hypothesis? And how do we know if we can reject one or the other? So we're not going to go into too much detail here, but I do want you all to remember that whenever you do any kind of comparison between data sets, and you can think of this in any kind of biological context, including phylogenetics, you're going to end up rejecting one or the other. And that usually is done through a p-value. So when biologists compare data sets statistically, what is the p-value? The p-value is essentially a probability value, and it can be defined as the probability that the null hypothesis is true given the data that you observe. So what p-value do you have to have in order to reject the null? Now, let's start really simply here and let's say that we're comparing whether patients respond better to a placebo or to some new drug that's been developed. All right, we want to see if patients really respond well to, say, some kind of asthma inhaler. All right. The null hypothesis is typically that there will be no significant difference in response between the placebo and the new inhaler drug. And you can apply that to any study that we're going to do. There will be no significant difference between the 2 trees that we're comparing, etc. The alternate is then the opposite. There will be a significant difference. All right. So if the p-value is defined as the probability that the null hypothesis is true given your data, that suggests that in order to accept the null hypothesis, your p-value has to be pretty high, right? The probability that the null hypothesis is true has to be high in order to accept the null. So what does the p-value have to be for you to reject the null? It has to be really low. And the cutoff is usually 0.05. A p equals 0.05 value, which is equal to 5%. So if your p-value is 0.05 or less, your null hypothesis is most likely not true, and so you're going to reject it. If your p-value is above 0.05, then you will tend to either accept or fail to reject the null. So keep those ideas in mind. And we're going to move forward with just these basic statistical ideas, okay? And let's talk a little bit about partitions, because this is what this topic is all about. we've talked already, or I have, and you guys have listened, about the difference that you can get in phylogenies when you're comparing two different genes. You can also get differences when you're looking at morphology and molecules, which is what we're going to do today. You can also get differences when you're comparing a gene tree to a species tree. And a species tree really is just the combination of all the different data that you put together to get a tree, a true tree of the species relationships. A partition is something a little bit different, but it applies to what we've been doing, and it can be defined as two or more independent sources of data that are used to estimate a phylogeny of the same set of taxa. So a partition might be morphology and molecules, or morphology and DNA. They're two independent sources. Now, they are dependent in terms of your DNA encodes for morphology, but we've coded them separately. All right, so a partition is two or more independent sources of data, say morphology and molecules, that you use to estimate the phylogenies of the same set of taxa. So remember that, it's the same set of taxa. So you're looking at two different data sets in order to determine how the same group of taxonomic units are related to each other. And there's a couple of things that might happen. Your partitions, say morphology and molecular data, might suggest either similar or identical evolutionary hypotheses, so their phylogenies might look exactly the same, or most likely, the partitions suggest different evolutionary hypotheses. Their phylogenies are different, and so which is correct? Is it the morphology? Is it the molecules? Is it gene 1 or is it gene 2? So a partition, just to review, is when you've got two different data sets. It could be morphology and molecular data, or it could be molecular data set 1 and molecular data set 2. What if two different molecular data sets of the same group of taxa are telling you two different things? What do you do in that case? How do you know which tree to accept? So those are the two different options. And just to show you what a partition might look like, this is based on DNA, of course, is you've got partition 1 and partition 2 of the same group of taxa, species A through H, and those different partitions are going to give you either a similar answer or two different answers. And so we're going to talk about how do you deal with those differences. Do you reject one of the data sets or do you accept both of them, right? So we're going to talk through carnivores. This will be our example that we use. And we're going to go back to parsimony because some of these methods that we're going to talk about that I will mention are based on parsimony, but you can also use them for maximum likelihood. In Bayesian inference, your posterior probabilities are actually a good test of whether your data are congruent or not. All right, so a Bayesian analysis does this automatically. So we're going to imagine that we have 10 in-group taxa, 10 in-group carnivores, and we've got one out-group, which is usually what we do with parsimony. And our first partition involves morphology. So we've got a couple of different critters on this slide. We've got a civet, which is the organism on the left, that mammal, and then we've got a walrus and we've got a bear. Looks like a grizzly. All right, and so you align your you code your data. This is your morphological data, so there really isn't an alignment, right? And you've picked your characters. They're independent characters, and you've used criteria to decide which characters are homologous. Remember all these little details from before. And you get these two MPTs. An MPT stands for most parsimonious trees. These two trees are equally parsimonious. Now, if you look closely at these trees, and they look a little strange, but you should be able to tell the relationships. They look strange because there aren't really branches on some of the taxa. Where do you see a difference in these trees? The only real difference that you will note will be, so I will circle them, and you can probably notice these yourself, is in this clade here between seal, sea lion, and walrus. There doesn't seem to be good agreement on the relationship between those three different taxa, right? In one, seal and sea lion are together as sisters, and in the other, a seal and a walrus ends up together. So these are two trees from morphology that are already disagreeing. And so typically what we do is we can build a strict consensus. This is typically what we do with parsimony analyses. Other than those three taxa, we know pretty well the relationships between the other carnivores. And one thing that I want you all to pay attention to, and this always blows my mind, is take a look at the most close relative between or of a cat. And we're talking about cats generally, but just take a look at that. I grew up in South Africa and I saw hyenas all the time, and to me, they look like a dog. But it turns out that cats and hyenas are actually sister taxa. Who would have known that? All right. So where do the two trees not agree? The only place where we see disagreement was with the seal walrus sea lion clade. All right. And what I want you all to remember here is that this tree right here is the one tree for morphology that kind of brings all of our morphological data together. So we're going to use this tree in a couple of slides here after we look at the molecules. All right, so this is the tree that summarizes the relationships based on one partition. So a strict consensus is what we are going to use to combine our morphological data. There is pretty good agreement except for that one clade. So this is the tree that we are accepting for our morphological partition. All right. Now, let's say that we do a molecular analysis and of the same group. This is what we're going to get, and we are going to also use a strict consensus. So this is a different partition. We have based this analysis on DNA, and we've used this weird gene called transthyretin 2, and there's 1,116 base pairs. Everybody should now know what that means. All right, so one gene that we've looked at. The tree length is 790 steps, and turns out that the length of the morphological phylogenies is also 790 steps. So these are pretty good trees. They have the same number of steps. And the two most parsimonious trees here with the molecular data is there's some disagreement again, right? Here, the seal walrus sea lion clade is pretty well worked out. And so that's this group right here. So we feel pretty comfortable with our molecules that these form a clade where walrus and sea lions are sisters. But when we look at the consensus, which this is, of the molecules, cat, hyena, and civet relationships are not very well worked out. So we are going to assume that this is our molecular tree based on parsimony. And if we put these two trees together, look at the differences. The outgroup, by the way, is a mole. All right. So for some reason in the book, they don't have the mole labeled as the outgroup, but it is the outgroup. So we've got a mole as the outgroup, and the morphology tree is on the left. This is our strict consensus again. So strict consensus. And the same for the tree on the right, which is our molecular tree. All right, so these trees are fairly different. Look at the placement, for example, of the otter relative to the raccoon. In our morphology tree, the otter is way over here, whereas in the molecular tree, the otter is actually sisters with raccoons. All right. So, oh, come on. So what do we do with this? This poses a potential problem, right? The trees for the two partitions disagree. Morphology and molecule data give us two different answers. So which is better? Which do we accept? So what this could mean is either that one partition's data are wrong. So the data have been coded wrong in the case of morphology, or our alignments have been done incorrectly. Maybe we used the wrong alignment software. Maybe we used the wrong assumptions when we looked at the molecular data. Remember, this is not maximum likelihood. We're talking about parsimony, and so no molecular models have been used here. Parsimony does not allow you to do that, which is one of its weaknesses. So either one partition's data are wrong, or the two partitions have a different evolutionary history and both partitions' data are correct. So which is it? How the heck do you decide what to do at this stage? So I'm going to introduce you to a test that will allow us to discern whether one tree is better than the other, or make us decide whether one tree, the molecular or morphological phylogeny, is the better tree. Which one do we accept? So there's a couple of solutions. I'm going to introduce you to two different tests that we can use for a problem like this. The first is going to allow you to determine whether two or more partitions' trees are significantly different. And the particular test that we're going to talk about is called the Templeton Test, or the Templeton Topology Test. And the second test will allow us to decide whether, if we have two different partitions or more, should we combine those partitions, and will that perhaps give us a better tree? When we combine two different partitions' data sets, we call that concatenating. So a concatenated data set is one in which two partitions' data sets are combined. So we're going to talk first about the first test. It's called the Templeton Topology Test. And the question here that we're asking about the two partitions might be, is a partition significantly more compatible with its own optimal tree than with another partition's optimal tree? Now, guys, on the slide, you will notice that the two partitions, these are just generic partitions. They're both DNA. But remember, the first partition that we looked at for the carnivore data set was morphology. All right, but this is just an example. The null hypothesis in a Templeton test is that the two trees for the two partitions are not significantly different. So we're going to assume that the two trees, or the two partitions' trees, are enough the same. And the alternate hypothesis is that the two trees are significantly different. But if you think about it, how do you determine whether a tree is significantly different from another? You also have to assign a p-value. So how do you do that with a tree? And so what the Templeton test looks like, and I've used a simple example. I've simplified my explanations here because this can get a little bit more complicated. If you really want to know the mechanism behind how the Templeton test works, I am happy to talk about it. Let me know if you're interested, and I will work through an example with all of you in our group chat, but really you don't need to know all the nitty-gritty details. So let's say that we've got a morphological data set of 5 taxa, or we can talk about the carnivores if we really want. And we've got a molecular data set. And for each of these data sets, we get a tree. And if you look at the morphological tree, species E and B are sisters. And in the molecular data sets, taxa E and C are sisters. And so we have to decide which tree here is better. Can we even do that? Or are these two trees significantly different from one another? Since there's disagreement, we have to use the Templeton test. If the two trees were exactly the same, you don't even have to do any of these tests. We know that there is one tree that is the most likely evolutionary hypothesis. So, how the Templeton test works is you are going to rerun your analysis based on morphology. But before you do that, you take the molecular tree that was created or devised by the program. You put that molecular tree in program memory. So the program that you're working in, which is usually PALP, that's a program that was used many, many years ago, and it's really the only program that can do this test. You put the molecular tree in program memory, and then you rerun the analysis based on the morphological data. So you keep the molecular tree in memory, which is the tree on the right, and you rerun the analysis with your morphological data set. The molecular tree that you're keeping in your program memory is called the constrained tree. So you rerun your analysis based on morphology. You attempt to see whether any of your morphological trees match the molecular tree in memory. And what the Templeton test is doing here is it tries to look at each character one by one and see how often do those characters differ in terms of the number of evolutionary events. So let's say that you've got character 1 that evolved once in the morphological data set, but twice in the molecular data set. You set those up in a table, and it uses those numbers to decide how close or different the match is between the morphological trees that you just found and the molecular tree that is in memory. All right? And what happens is the Templeton test spits out a p-value. Now, based on what we talked about earlier, what I mentioned in terms of p-values is this p-value significant or not? Would you reject the null hypothesis or not? See if you can answer that, and I'll be quiet for a couple of seconds, or you can pause the video. Is this p-value significant? All right, if you convert this p-value to a percentage, it is 11%. And if you remember, the p-value has to be 5% or 0.05 or less in order to reject the null hypothesis. All right, so let's do the same with the molecular data. You put the morphological tree in program memory this time, and that is your constrained tree. Then you rerun the analysis based on the molecular data, and then again, you look to see whether any of the morphological trees or any of the molecular trees that you are just now getting match the morphological tree that you have in memory. How close or different is the match? How many times do the characters evolve in the molecular data set as compared to how those characters appear on the morphological tree? And let's say that we get a Templeton test here of 0.0008. Is that value significant? Would you reject the null hypothesis? You can pause, and I would argue that yes, you do reject the null hypothesis. So what does this mean exactly, right? You do not reject the null with the morphological data set, but you do reject the null hypothesis for the molecular data set. What that means is that both partitions, morphology and molecules, could have evolved a similar tree topology. And that basically comes from the fact that the morphological data, if you look at the morphological data set and that p-value, are not significantly different from the molecular tree. Remember that you ran the morphological data set, and then the trees you got from the morphological analysis was compared to the molecular tree that you kept in memory. Since the p-value is not significant, you cannot reject the null hypothesis, which means that the morphological data are not significantly different from the molecular tree, which means that you could still get the molecular tree using the morphological data. So the Templeton Test is used to determine whether you have enough agreement between your morphological and molecular data set to warrant accepting one tree over the other. What you would do then in this case is you would accept the molecular tree as the true tree and the most likely tree, the most parsimonious tree that best fits both data sets, because the morphological data set did not reject significantly the molecular tree. So think about that. Write down any questions that you have. Here is another example. Let's imagine that you have two genes, and I've used the same trees in this case. You can have two different genes that are also giving you two different phylogenies. Here we're going to do the same thing. Pardon the order of those 2 points, but first you're going to put gene 2's tree, in program memory, and you rerun the analysis based on the gene 1 data. Do any of the gene 1 trees match gene 2's tree in memory? And how different or close are those matches? And let's say that the Templeton test gives you a p-value of 0.034. Without even looking at gene 2's data, what is this telling us? What is this telling us? This suggests that there is a significant difference between the gene 1 tree and the gene 2 tree, right? We don't get the gene 2 tree when we are rerunning gene 1's data set. If we do the same for the second gene, by putting gene 1's tree in program memory, rerunning the analysis based on gene 2's data, and then comparing gene 2's trees with the program tree in memory, which is gene tree 1, we get a Templeton test of 0.002. Shoot, is that value significant? Yes, it is. So what this is telling us is that the trees that you get by running gene 2 again are significantly different from the tree that you get from gene 1. So what this suggests is that these two data sets yield two different trees that are significantly different. They are telling two different stories, and neither data set will accept or match well enough a tree of the other data set. It suggests that these two partitions are significantly different from one another. What do we do now? So we are going to go into the next test, which we can use for a couple of different reasons, but let's imagine that this is one of the scenarios that we end up with. What do we do now? Right? Which gene tree's data is correct? Luckily for the morphology and molecular data set that we just looked at, you can accept the molecular data set or the molecular tree, because the morphological data set was not significantly different from the molecular tree.